We started imaging here in Maine because UCI has an outstanding expertise in biomedical imaging, in particular in areas like confocal fluorescence microscopy. Imaging data is big data. It contains a huge amounts of dense, useful information. And that can really be helpful in explaining some of the underlying biology. So the temporal and spatial dynamics are really key to understanding that. So it's natural that when modern workflows in spatial omics, like transcriptomics, proteomics, and genomics, that can access imaging data down to the cellular or subcellular level, it's natural that the one part that's missing there is the metabolomics and lipidomics. And mass spec imaging is the way to basically address those questions. We have a number of projects that are ongoing at the moment. We've had the Shimadzu iMScope now in operation for about a year, and about a dozen research groups that are using it on a routine basis. So those types of projects include the neurobiologist, many groups looking at dementia diseases like Alzheimer's. We have a cancer center with groups looking at metabolites and lipid disturbances in solid tumors. Plant biologists looking at plant metabolome changes, particularly pigment synthesis. Groups from engineering that are looking at artificial cartilages and the distribution of collagens. One particular specific project I'd like to highlight is from the Department of Urology. So that's work by Michael Wu, Jamie Landman, and Ralph Klayman. And they're looking at urinary tract diseases such as bladder cancer and urethral cancer, which is very difficult to treat because getting the drugs into the tissue is quite challenging. Their model is to use a catheter to deliver the drugs in the lumen, but then use low pulse voltages to deliver the drugs into the tissues. And using the iMScope, we've been able to show that the drug moves in a directional way into the tissues and accumulates by more than 10-fold. One of the benefits of the iMScope is that we're able to generate good calibration codes with about a 200-fold linear dynamic range using stable isotope analogs of the drugs incorporated into the matrix when we apply it. I think the benefits of the iMScope are basically threefold. One of the major benefits is the built-in optical microscope. That's really useful because that allows us to very easily register the sample to the optical image that's generated within the instrument. Consequently, the workflow is very much simplified for the end user. Second part in the workflow here is also setting the instrument parameters for the data acquisition. And the interaction between the user and the software is also very intuitive and straightforward. There's only a few parameters that you really have to consider. It's the laser frequency, the laser power, the pixel pitch between the spots on the image. And so once you've set that, you can store those settings for reuse or for optimization of the conditions. As a consequence, we can get a user to go from sample introduction to a data acquisition within 5 minutes. And that's really, really helpful in a high throughput setting. A second major benefit is the speed with which the instrument acquires data. So the Shimadzu iMScope has a 20 kilohertz laser. As a consequence, we can run the instrument at up to 40 to 50 pixels a second for data acquisition. And that's really critical, especially in a high throughput facility like ours when we have lots of users. It also means that we're not constrained by going down to very low spatial resolution. A lot of biologists want the highest resolution data possible, which typically means down to single cell level, around about 5 micrometers. At that spatial resolution, most imaging systems that are running at only a couple of pixels a second would take way too long, making them impractical. The iMScope doesn't have that limitation by being able to run at a much higher data acquisition rate. A third key difference is that the iMScope operates as an atmospheric pressure MALDI, which is in contrast to the high vacuum MALDIs found in many other systems. So we have noticed a few differences in ionization, but importantly not down in the lower mass ranges below 1500, which is relevant to metabolite and lipid signaling. The atmospheric pressure MALDI therefore has the ability that we can put a sample in and acquire data without having to pump down and wait for the instrument. That's one key benefit. Another one is that because it's atmospheric pressure, we don't run into problems with matrix sublimation, which we often get under high vacuum systems. And that allows us to expand the repertoire of matrices that are usable in the iMScope, and particularly potentially also for very exotic matrices like ionic liquids. I think there's several areas where the field of mass spec imaging will advance. The first is basically the development of new matrices that are better tuned to absorb laser energy for ionization efficiency, and also to control the crystallization process. The finer and smaller the crystals and more homogeneous, the better the spatial resolution that we can attain. So nitroindole derivatives are one class of new matrices that look quite promising in that regard. And hopefully that will boost sensitivity. So sensitivity is really a key factor here in mass spec imaging. A lot of the interesting biology is down at the very low signal to noise level, and we often can't get sufficient data quality at that point. So any boost in the signal to noise or sensitivity will bring those molecules into focus, which will allow us to expand the type of biology questions that we can answer. I think another key area in advance is using MALDI imaging for proteomics. That's been a challenge because the traditional workflow is to take FFPE tissue, delipidate it, and then do antigen retrieval, followed by in situ trypsinization to release de novo peptides that can be imaged. During that process, you lose a lot of sensitivity. You often get partial digests, or you can delocalize the signals. One promising approach to get around that is the use of multiplex MALDI immunohistochemistry, where you can use photocleavable mass tags, so basically antibodies coupled to a photocleavable linker with a specific peptide mass. And that allows us to use multiplex approaches, looking at many different proteins simultaneously. But the important thing here is that because it's a photocleavable event, we get 100% cleavage, we don't lose signal sensitivity, and we retain the image localization. So that circumvents a lot of the problems that we see with the traditional proteomics workflows. And I think that's a really exciting area in mass spec imaging that will expand the use of this technology. An additional advance that I think is coming in the field of mass spec imaging is the software, the data analysis. Imaging data is very complex, but one of the benefits of mass spec imaging is that we collect data on dozens, if not hundreds of different species simultaneously and in the same concurrent image. So that opens up the possibility of looking at correlation analysis. So each pixel can be correlated to each other one, and each individual component can be correlated. That allows us to segment and classify specific markers within tissues, and that can be very, very helpful in understanding the underlying biological processes. So having software that can do that in a simple way is a really key aspect. The Shimadzu ImageReveal software is ideally suited to that. It has a very intuitive, very linear, simple organization, yet very powerful tools behind it. I think in the future, advancements here in the imaging algorithms using artificial intelligence or guided approaches to the data analysis would be very helpful. That and the identification of the specific metabolites is really the key limiting factor at the moment that makes it a little bit challenging for a regular user to get the most out of the data. And I think having software that addresses those questions will be a really big benefit to the field.